I'm Francesco Girardi, and before starting, I would like to really thank the organizer to give me the opportunity to present here and to finally be back here in a muscle meeting. I'm super happy to meet again many friends and colleagues of mine. Today, I'm here actually in a totally different role. In fact, I'm now application specialist from Media System Lab. We are a microscope distributor company, and today I would like to introduce you to two live cell imaging technologies. One is Orox, the other one is NanoLive. Orox is a laser-free confocal microscope, while NanoLive is a holotomographic microscope. Both of them are perfectly suited for live cell imaging, so let's see how these two technologies could kill the imaging stars. Before starting with the actual presentation, let me just give you a brief introduction on live cell imaging. We all know that biological processes are extremely complicated and dynamic, with several subcellular interactions and many interactions between cells. All this is happening in 3D and during time, so having a complete picture of all these events is essential to better understand the process of interest. Unfortunately, with conventional microscopy, it's really hard to achieve this task because we are always using fixed sample that will provide us only a single snapshot of the reality. It is here that live cell imaging becomes a valid alternative to the traditional technologies. However, live cell imaging is not that easy to perform. In fact, the main issue with live cell imaging is the cell wellness. We have to keep our cells alive. Obviously, if we do not have a proper incubator under our microscope, our cells will simply die. That's basic. However, the main issue with live cell imaging is related to fluorescence. It's called phototoxicity, and by using fluorescent dyes, lamps, or lasers, we are impacting our sample, and of course, it will produce reactive oxygen species until, boom, they explode. Both these movies were recorded by me during my PhD, so as you can see, I did not have that much luck with these technologies. It was a real struggle, but today I'm happy that I'm here to present you two amazing solutions for these kind of problems. The first solution I have for live cell imaging is the 3D Cell Explorer from NanoLive. NanoLive, as I said, is the first holotomographic microscope in the world, and it allows you to visualize your cells in 3D and in time-lapse without any fluorescent staining. All this is a non-invasive technology at a high resolution and a high sensitivity. Moreover, as you can see from the picture, NanoLive is a very little and compact microscope provided with a little top stage incubator, so perfect for live cell imaging. Let's see how it works. As I said, it's a holotomographic microscope, so it's a new word. It combines holography and tomography to obtain a 3D map of the refractive index of the sample. So with holography, we are referring to a very gentle laser, 520 nanometers, that will go through the sample and obtain a 2D map of the refractive index of this sample. But now, how can we pass from 2D to 3D? Is thanks to tomography. Thanks to this rotating arm, the laser beam will hit the sample at 360 degrees. So from 2D, we make all the circle, get a 360 hologram of the cells. And if we combine them all together, this is a classic result of NanoLive. Here, thanks to the high resolution, we are at 200 nanometers X and Y, and thanks to the high sensitivity, we can reach one image every 2 seconds. The detail, the subcellular detail of the cells are amazing, even if we are not using any staining. We can see the nucleus, the nuclear membrane, all these worms moving inside the cells are the mitochondria, and all the dots, white dots, bright dots, are the lipid droplets and all the vesicle. Again, I would like to underline that all these movies are in 3D, so you will be able to even get the rendering of your process. In this case, we have a beautiful mitosis, and with all together in this slide, what we can appreciate is that NanoLive can really provide beautiful movies without any staining. Removing fluorescence from your experiment brings two great advantages. The first one is zero sample preparation. You plate the cells and you're ready to go. The second one is zero phototoxicity. As I said, phototoxicity is the main issue with live cell imaging, but with NanoLive you will eliminate phototoxicity and you will be able to visualize any kind of cell culture in vitro with no time limits, both in length of the movie and in frequency. So, said that, the application of NanoLive, at least in vitro, are infinite. We could bring here many examples, but since we are in a muscle meeting, I decided to show you some muscle experiments. The first experiment is a very easy experiment that one of our clients performed, and those are human skeletal muscle cells differentiating, so they are already on myotube. Here, just to show you that we can still see within the cells, so we have a high resolution at the cellular level and see all the dynamics. Of course, this is a very simple experiment, it was just a test. But imagine by looking at the entire population, checking all the fusion process. I mean, I'm interested on that because I was working on this, but I will suggest everyone to try that. Another example, still in muscle, I wanted to show you are those beautiful cardiomyocytes proliferating. If you follow this one, it will divide. What I like about this movie is that it's not shaking, but the cardiomyocytes are actually beating. I show this example because I would like to underline again that these cells are totally fine. There are no bias introduced by NanoLive, again, because it's a totally label-free technology, and the cells are free to live. This is a 6-hour movie, one image every 30 seconds. They are totally fine. Changing topic, we are moving into the immuno-oncology, mostly the main topic with NanoLive. Here I'm showing you a co-culture with cancer cells and cytotoxic T cells. So if you follow the red cell, the cancer cell, so bigger and spread, you'll see that poof is dying of apoptosis induced by the T cells. What I want to show with this example is that with NanoLive you can actually perform cell population dynamics, so see the entire interaction between this population, but still have the high resolution at the cellular level, so distinguish even the kind of cell death. However, I've been a researcher as well, and I know that at certain points beautiful movies are not really useful. We want something concrete. So how can we translate these movies into something real, like a result? It thanks to the analysis software that comes with the microscope. NanoLive produces EVE Analytics. This software will allow you to automatically analyze the movie you're performing. They're changing color because with a very easy two-step segmentation, the software will be automatically distinguish the cells from the background and automatically provide you 12 parameters. This is just the basic version of the software. There are plenty more parameters. They are very simple parameters like cell count, confluency, area and perimeter. There are more complicated parameters like granularity and dry mass. But altogether, they will allow you to analyze directly and automatically your movies, providing you information about cell morphology, cell population, and cell content. Okay, concluding with NanoLive, here I'm showing you the free configuration of this microscope. What I just presented was the CX, so the basic one, the technology. But in the last year, it has been upgraded with two great advantages. The first step was the integration of fluorescence module. So with CXF, you can actually perform holotomography together with fluorescence. But the main instrument from NanoLive now is the CXA that has the fluorescence module and also a motorized stage. So CXA is the only holotomographic microscope in the world that will allow you to perform multiple samples simultaneously, up to the 96 well. Okay, so this one was the first solution I had for live cell imaging. The second solution I wanted to show you instead is coming from Orox, London, and is called Unity. Unity is a laser-free confocal microscope. And as you can see from the picture, again, is a very little and compact microscope. Within this black box, you have a full microscope. You have the camera, the light source, the objective, a spinning disc, and the red part is an incubator with a motorized stage. So perfect, again, for live cell imaging. As I said, it's a laser-free confocal microscope, so let's see how it is possible that a confocal microscope is not using lasers. Orox patented a very particular spinning disc. Instead of having the classic pinhole distribution in spiral, is replacing the pinholes with a grid scan, with a grid pattern, so a rectangular slot. While with the conventional spinning disk, only 1% of the light can pass through the disk, so we need powerful light sources like lasers. With Orox, up to 50% of the light can pass through the disk, meaning we are able to use even more gentle light sources like LEDs. So using LEDs provides two main advantages. The first one is money related. They cost less than lasers and they have no maintenance. On the other side, instead, we have also technical advantages, which are related to phototoxicity. LEDs are, in fact, producing less damages to the cells. So in conclusion, with Unity is a LED spinning disk, but even if it is perfectly suited for live cell imaging, you can still consider it as a classic spinning disk. Thanks to the motorized stage, you'll be able to perform multi-positioning, mosaics, Z-scan is compatible with all labwares and so on. But all this in a very user-friendly way. In fact, instead of having a software, Unity is controlled via iPad. Instead of having a software, they have an app, and in only one page, you'll be able to set up your experiment. That's very easy. Okay, so my two solutions for live cell imaging are actually concluded. I presented them. I just want to spend the last minutes talking about Clarity, which is the second product from Orox. Just to tell you that if you like this structured illumination technology, Clarity is an add-on that you can attach to your own wide field microscope and transform it into a spinning disk. So we're talking about a totally different instrument, but it shares the same technology. So it has the camera, the LED, the spinning disk, the filter, and you attach it into a wide field microscope or a broken confocal microscope, and you give a second life to your microscope. Okay. To show you the improvements that Clarity can bring to your own wide field, I choose again a muscle cryosection, a muscle example for this meeting. On the left side we have the wide field, on the right side we have the confocal. I'm not here to explain you the differences between spinning disk and wide field, but of course I just wanted to show you the muscle cryosection. Okay, so actually my presentation is concluded. As Media System Lab we have also a third microscope, but is a 2-photon microscope. So since we are not really in a neuroscience conference and is a very complicated technology, I decided to not present it. However, if you're interested, we also have Fentonics. Fentonics is a Hungarian company that produces Femto 3D Atlas. It's the one and only acousto-optic 2-photon microscope. To make it simple, because I don't like this physicist stuff, I like to make a biological example. If with conventional microscope we can have 2D live imaging in vivo, with Femto 3D Atlas we can have an ultra-fast 3D imaging in vivo. So that is a very complicated technology. If you have any question, please come to me. But those are my contacts. I will be around here today. So if you have any question, I'm here available to talk about those. Thank you very much.
Okay, questions? Well, if not, you are available here today, tomorrow?
Only today.
Only today. So
be quick.
Be quick. First question. Okay. There's a question? Yeah, she has a question.
Tissue staining, things like that?
If you're referring to nanolive, no, because it's actually for 2D cell culture. So very simple sample, only because having a very gentle laser to avoid phototoxicity, he has very low penetration qualities. Yes, indeed. With organoids, you can check the surface of organoids, for example, but not inside, but all around you will be able. With Unity instead, which is a spinning disk confocal microscope, in that case, you can go through. Also because LEDs are less scattered light compared to laser, so you can go deeply in the sample. In that case, you'll be able to go inside. Thank you.